I would like to welcome you to the Department of Plant Systems Biology at the VIB University of Ghent, and specifically to the Systems Biology of Yield group headed by Professor Dr. Dirk Inzee. Here we apply systems biology approaches for studying yield phenotypes in plants. Our recent manuscript that was published in Developmental Cell highlighted the dynamic processes of early leaf development in Arabidopsis. Here to tell more about the findings is Megan Andriankaja.
So Megan, can you tell us why you started this project and what you found?
Actually, what we'd seen previously is that many of the genes responsible for enhancing leaf size are acting on the process of cell proliferation, and specifically the duration of this cell proliferation. So we wanted to further characterize these crucial early stages of leaf development when cells in the leaf are transitioning from proliferation to expansion. And what we found was that the transition from proliferation to expansion in the leaf happened much more rapidly than previously thought. And even more surprisingly, we found that these rapid changes in proliferation were very tightly coordinated with the onset of photomorphogenesis, and that by blocking chloroplast differentiation, we could also inhibit the onset of cell expansion.
How did you visualize this transition of the cells from proliferation to expansion?
We wanted to look at both the morphological changes in the leaf as well as the molecular events. So we took a dual approach. First, we profiled the leaves at the cellular level phenotypically by drawing the abaxial epidermal cells of the leaves, and then we also did a transcriptome profiling of leaves at the same time points. So for the cellular analysis, we used a differential contrast microscope that is fitted with a special drawing tube that allows us to simultaneously visualize our leaf mounted on a microscope slide, and then we overlay this onto a drawing area so that we can trace the outlines of each individual cell in the epidermis. So we drew these cells along the entire length of the leaf, and then representatives of these drawings are shown here. These drawings were then analyzed using in-house image analysis algorithms, which allowed us to measure the area, the shape, and the position within the leaf of each individual cell. In the end, the size and shape criteria were used to define a cell as either dividing or expanding, and the positional information allowed us to plot where in the leaf these dividing and expanding cells were located.
And how did you go about the transcriptional profiling of these small leaves then? Because I assume a dividing leaf is still a very small tissue.
So what we had to do is we had to microdissect these small leaves in very large quantities in order to get enough material for a tiling array hybridization. And what we found from these analyses is that many of the things we'd already seen from the phenotyping were also found back in the transcript data, such as non-cell proliferation genes being downregulated and cell expansion genes being upregulated over the time course. However, what we also found was a number of genes involved in photomorphogenesis, and these were tightly co-expressed with the cell proliferation genes. So we postulated that these two processes, the onset of cell expansion and the onset of photomorphogenesis, were tightly coordinated. And more specifically, we also saw transcripts responsible for chloroplast differentiation induced just prior to the rapid shutdown in cell proliferation. So we hypothesized that some retrograde signal from the chloroplast to the nucleus was responsible for regulating when these cells in the leaf ceased proliferating and began expanding. We tested this hypothesis by blocking chloroplast differentiation using a carotenoid biosynthesis inhibitor, norflorazone. And what we found was that cell expansion was inhibited when we blocked chloroplast differentiation, which indicated that these leaves do need properly functioning chloroplasts in order to begin expanding.
That's great. So what do you think is the significance of these findings in the field of leaf development?
We believe that these early stages of leaf development, when cell proliferation is most active, these will be critical in determining the final size of an organ and thus its biomass potential. So this research really provides us a baseline and a strong framework for all future studies on early leaf development, especially at these crucial time points in the earliest stages of the leaf development.
Okay, thank you, Megan. That's really interesting, and I look forward to reading your paper in Developmental Cell.